we are going to talk about the digestive system, which is Unit 8. This
And with her new teeth, she can take on more solid foods. Digestion starts in the mouth. Teeth grind up the food. Then, special glands under the tongue pump out saliva to help break down and lubricate the food on its 12-hour, 13-foot journey through the gut. It'll pass from the stomach into the coils of the small intestine before finally passing into the large intestine. Waves of contracting muscle keep the food moving, a process called peristalsis. These contractions are so powerful, we can even eat upside down. For the first time, a new camera shows a high-definition view of how food travels through our bodies and into our stomachs. Food enters the stomach through a hole at the top. The stomach is a bag of muscle that churns, squashes, and squeezes food into liquid. At the same time, acids break the food down. The stomach walls protect themselves with a lining of mucus. Without it, the acids could digest parts of the stomach itself, causing stomach ulcers. About an hour later, the stomach squeezes the broken down food out through a tiny hole called the pyloric sphincter. The food enters the small intestine, an 11-foot coil of tube where we absorb most of the nutrients. The interior wall of the small intestine is lined with millions of microscopic projections called villi. These increase the surface area of the gut, making it easier to absorb nutrients. First, the pancreas pumps out a juice that neutralizes stomach acid. Then bile from the liver breaks down the fats into tiny droplets. Smaller droplets are easier for the intestine to absorb. After an hour and a half, the small intestine has absorbed most of the nutrients from the food. It's time for what remains to move on. It enters the large intestine through this, the ileocecal sphincter, a valve that keeps our food from going back where it came from. What's left is a mix of waste food and dead cells from the walls of the gut. The large intestine's main job is to extract water from it. Lots of bacteria live here too, but it isn't because of an infection. We actually need them. They produce enzymes that break down complex carbohydrates in our food, carbohydrates we couldn't otherwise digest. Finally, after about 12 hours, we expel what's left of our first meal.
So that kind of gave you an idea of what we're going to be talking about today, the digestive system. And now in the digestive system, the organs within that system can fall into two groups. The first is the alimentary canal, and this is a continuous muscular tube. And this is going to include a few of the items on here. Of course, the alimentary is going to be the mouth or that oral cavity. Next you're going to see is the pharynx, the esophagus, the stomach, the small intestines, which you can see subdivided there, and we're going to talk more about those later, and the large intestines. And basically the main function of that area is to digest and absorb food. So just think, it's that one that follows through that tubing all the way around. The next is basically the accessory digestive organs. Those are, it's not here, but it's the teeth, right? The tongue. There are salivary glands that are located. So all of those are in the mouth. You also have the gallbladder. You have the liver. You have the pancreas. And basically, all of these guys are there to aid in mechanical digestion, mechanical breakdown, and they all help to either break things down mechanically or they help to produce specialized secretions. And these secretions aid in digestion, right? And so all the organs within the body can fall into one of these two groups. Now there's three key concepts that help to regulate GI activity. The first is the digestive activity is provoked by a range of mechanical and chemical stimuli. So when we're talking about this, you basically have receptors that are located in the GI tract organs. So those are one way that it can basically determine if digestion needs to happen. Another way that mechanical or chemical stimuli can be picked up is it can happen, or you can get digestive activity happening because of things like stretch or changes in osmolarity or pH or presence of different molecules. So all of those things can help to provoke digestive activity. Now the next key concept is that the effectors of digestive activity are basically smooth muscle, and glands. So when stimulated, the receptors that we were just talking about can initiate reflexes that actually stimulate smooth muscles. And so those smooth muscles are there to help mix and move the content of those that's within that alimentary canal. Now, at the same time, reflexes can also help to activate or inhibit digestive glands. And these glands help to secrete digestive juices or hormones. And the third that we have is the third concept that we're going to hit on is that you'll have neurons, both intrinsic and extrinsic, and hormones that help to create or help to control digestive activity. So the intrinsic controls, these are short reflexes, and we're going to talk about those being regulated by the enteric nervous system, which is within the actual digestive system itself. And then when we're talking about extrinsic, these are going to be longer reflexes, and those are going to be autonomic. And we're going to go into detail about those. When we're talking about hormonal control, hormones from cells in the stomach or small intestines can actually target cells in the same or different organs to secrete or contact. And we're going to talk a little bit more about how the hormonal controls work. So there's four main functions of the digestive system itself. The first is to take in food. Obviously, we do that through the entry of the oral canal. Next, what we're going to do, or the digestive system is going to do, is it's going to break it down into nutrient molecules. Once we break down those nutrient molecules, we want it to get into the body. So we're going to absorb those molecules into the bloodstream. That's our highway to get throughout our system. And anything that we don't need, obviously, we're going to rid our body of any indigestible remains. And so those are our four main functions of the digestive system. So now we looked at the digestive system, and I showed the first video to kind of give an overview and so that you could see what it looked like inside of every single organ. I thought that was really neat. But this is going to give us a general overview from start to finish, what's happening in the digestive system. And then what we're going to do is we're going to pull each part separately and talk about basically the process of breaking down food, how food moves through, how we enter it, how mechanical digestion works, how chemical digestion works, and then all the different organs.
Across the whole planet, humans eat on average between 1 and 2.7 kilograms of food a day. That's over 365 kilograms a year per person, and more than 28,800 kilograms over the course of a lifetime. And every last scrap makes its way through the digestive system. Comprised of 10 organs, covering 9 meters, and containing over 20 specialized cell types, this is one of the most diverse and complicated systems in the human body. Its parts continuously work in unison to fulfill a singular task, transforming the raw materials of your food into the nutrients and energy that keep you alive. Spanning the entire length of your torso, The digestive system has 4 main components. First, there's the gastrointestinal tract, a twisting channel that transports your food and has an internal surface area of between 30 and 40 square meters, enough to cover half a badminton court. Second, there's the pancreas, gallbladder, and liver, a trio of organs that break down food using an array of special juices. Third, the body's enzymes, hormones, nerves, and blood all work together to break down food, modulate the digestive process, and deliver its final products. Finally, there's the mesentery, a large stretch of tissue that supports and positions all your digestive organs in the abdomen, enabling them to do their jobs. The digestive process begins before food even hits your tongue. Anticipating a tasty morsel, glands in your mouth start to pump out saliva. We produce about 1.5 liters of this liquid each day. Once inside your mouth, chewing combines with the sloshing saliva to turn food into a moist lump called the bolus. Enzymes present in the saliva break down any starch. Then your food finds itself at the rim of a 25 centimeter long tube called the esophagus, down which it must plunge to reach the stomach. Nerves in the surrounding esophageal tissue sense the bolus's presence and trigger peristalsis, a series of defined muscular contractions. That propels the food into the stomach, where it's left at the mercy of the muscular stomach walls, which pound the bolus, breaking it into chunks. Hormones secreted by cells in the lining trigger the release of acids and enzyme-rich juices from the stomach wall that start to dissolve the food and break down its proteins. These hormones also alert the pancreas, liver, and gallbladder to produce digestive juices and transfer bile, a yellowish-green liquid that digests fat, in preparation for the next stage. After 3 hours inside the stomach, the once shapely bolus is now a frothy liquid called chyme, and it's ready to move into the small intestine. The liver sends bile to the gallbladder, which secretes it into the first portion of the small intestine, called the duodenum. Here, it dissolves the fats floating in the slurry of chyme so they can be easily digested by the pancreatic and intestinal juices that have leached onto the scene. These enzyme-rich juices break the fat molecules down into fatty acids and glycerol for easier absorption into the body. The enzymes also carry out the final deconstruction of proteins into amino acids and carbohydrates into glucose. This happens in the small intestine's lower regions, the jejunum and ileum, which are coated in millions of tiny projections called villi. These create a huge surface area to maximize molecule absorption and transference into the bloodstream. The blood takes them on the final leg of their journey to feed the body's organs and tissues. But it's not over quite yet. Leftover fiber, water, and dead cells sloughed off during digestion make it into the large intestine, also known as the colon. The body drains out most of the remaining fluid through the intestinal wall. What's left is a soft mass called stool. The colon squeezes this byproduct into a pouch called the rectum, where nerves sense it expanding, and tell the body when it's time to expel the waste. The byproducts of digestion exit through the anus, and the food's long journey, typically lasting between 30 and 40 hours, is finally complete. TED-Ed is a nonprofit.
So that kind of gives an idea of how it works. And so let's go into processing of food. It actually involves 6 essential activities. As you can see, the first is ingestion, and it's right there. So ingestion is a process of actually eating, taking food into the mouth, right? And then number 2, whenever we're talking about this, we're actually talking about propulsion is the second. And so this is the propulsion is the actual movement of food. And this happens or involves it going through the alimentary canal. So you can see here, you're going, and it's talking about the movement going through that tubing there. So it's composed of basically two parts that we're going to focus on here is swallowing and peristalsis. And so swallowing is just this idea of making it so that it separates, kind of forms its vacuum to allow it to start going down through the esophagus. And then peristalsis is the major means of moving food or propulsion of food. And basically it's going to involve alternating waves of contraction and relaxation. Now the third activity or major process that's occurring during the digestive system is mechanical breakdown. You can see that up here. And so mechanical breakdown is going to include in here chewing, mixing food with saliva, churning the food in the stomach, and this process called segmentation. Don't get too concerned about understanding the difference between peristalsis and segmentation because I'm going to hit on this after this. And basically segmentation is local constriction of the intestines that helps to mix food with the digestive juices. And so it's basically shown you this all up here: chewing, churning, segmentation. Now the fourth part is digestion. And so digestion here, I'm going to move this down here so we can just see it a little bit better, is a series of catabolic steps. And we know catabolism is the process of breaking down. And this takes enzymes to do, right? And so steps, and so basically what's happening here is that we're taking complex food and breaking it down into chemical building blocks. Now the fifth part is here, which is absorption. And basically this is the passage of food from the lumen of the GI tract to the blood or lymph. And defecation, which is down here, the sixth part, that's the elimination of indigestible substances. They called it stool. Here, the term they also use is feces. And so this is how food is processed, and these are the 6 essential activities. So what we're going to talk about now is kind of the difference between peristalsis and segmentation and how this affects those activities. So peristalsis, this is when we were talking about propulsion, number 2. This is the main way of moving. So when you chew, the food in your mouth, it's going to mix up with saliva, and it's going to make this thing called a bolus. And this bolus is kind of this round, packed, oops, sorry guys, entity here, right? And so when you swallow and it starts to go into your esophagus, what will happen is that your esophagus is going to contract. And when it does, it's going to push this guy forward. Now, as you can see at this point, this area that was originally contracted is loose, and the contraction happens farther, pushing it farther down. So this is what's happening. It's basically contracting just farther and farther, and it's going to push it until it gets primarily through the esophagus or here. So the point of peristalsis is primarily for propulsion. There's some mixing that can occur, but it's pushing it through. Now, segmentation is different. If you see, this had adjacent segments. So you have this, and then the adjacent segment that's right below it contracts, and then this. But here is different. Here is adjacent segments of the canal will contract. So if you see here, you have the bolus. And what happens is it contracts right here. And so you can see it's pushed down here. So what happens is that bolus, or that gets mixed. Half of it goes here, half of it goes here. And then as you can see, these guys contract here and mixes it back together. So this is basically mostly for mechanical breakdown. You're pushing it. It's kind of like having having some kind of bag and pushing stuff in to kind of mix it in the bag. And you're pushing down on different parts of the bag, and as you do, everything kind of mixes together. That's what segmentation is. And this happens mostly when you're looking at it in things like the small intestines. So that's the difference between peristalsis and segmentation. Now, as we go through the digestive system, it is connected, or basically, the GI tract essentially has 4 different layers. And it's normally surrounded by what's known as the peritoneum. So the peritoneum both has the visceral and the parietal peritoneum. If you remember, when we were talking about serous membranes, this is somewhere on that, right? So the visceral peritoneum actually covers the external surface of most of the digestive organs. The parietal peritoneum lines the body wall. Of the abdominal pelvic cavity. And you'll remember we learned about that in A&P1 when we were learning about serous membranes. The peritoneal cavity is located between these guys. And it's filled with serous fluid. So basically it's helping to reduce some of the friction, because as you can tell, just by looking at some of the videos, there is a lot of movement that's happening from the organs of the digestive system. Now the mesentery, which recently they've decided is its own organ, is a double layer of peritoneum. And it extends to the digestive system from the body wall. And so what this is going to do, it's going to allow blood vessels, lymphatics, and nerves to reach the digestive system. It also helps to hold the organs in place, and it stores fat, right? So it has three main functions, right? So blood vessels, lymphatics, and nerves to reach the digestive organs, right? 2, keeps organs in place. And 3, stores fat. So that's the mesentery. Now, the types of organs you have are the intraperitoneal organs and the retroperitoneal organs. So the intraperitoneal organs are organs that are located within the peritoneum. And the retroperitoneal organs are organs found posterior of the mesentery. And so they're lying against the abdominal wall. So in here, you include mostly like the pancreas, the duodenum, and parts of the large intestines. So let's kind of look at this, at the different parts here. So most digestive organs are the intraperitoneal, and they're suspended from the body wall by a dorsal mesentery, right? And so if you look here, right here is the peritoneal, parietal peritoneum, and then this right here is going to be the visceral part, and then if you look in the middle of all that is the alimentary canal organ. And so most of them are going to fit in this way, and that's the intraperitoneal, if they fit in there. Some intraperitoneal digestive organs are also suspended from the body from the ventral. So this is if they're suspended from the dorsal. This is the ventral. So a perfect example of something like this would be the liver, right? And then some digestive organs are called the retroperitoneal because they have basically they've lost their mesentery during development, right? So they're on the outside here. And if you're trying to figure out what the basically the mesentery looks like, you are going to look at this area that is connecting the small intestines. All that area that you're looking that radiates and kind of keeps it in place. That's what the mesentery is. That's what we're talking about here. Now, when we're looking at the alimentary canal, all digestive organs have the same basic layers, or those layers are called tunics, right? We have the mucosa, we have the submucosa, the muscularis externa, and the serosa. And so we are going to actually look at those basic, and we're going to look at them in the lab as well. So let's look at the basic structure here. And so the most outward layer, the number one that we're going to look at is the mucosa right here. And you can see that mucosa, essentially, This is the innermost layer. And so when we talked about here, we're going from inside the innermost layer to outside here. And so let's start on the innermost layer. So if you see here, here's the lumen. That is basically where all the digestive substance is going to be here. So the innermost layer is that mucosa, and you can see it's made of the epithelium lining that's going to be right here on that inside, right? The next is going to be that lamina propria, and then you're going to see within that the muscularis mucosa. And so when we look at that first layer, this guy right here, it has simple columnar epithelium, if you remember back from the histology section when we're talking about epithelial lining and where certain parts are located, right? It has basically the mucus secreting cells here. And this is basically where you're finding is in the mouth, esophagus, and the anus. They are going to be made up of mostly stratified squamous versus simple cuboidal. Again, this area helps. It is the mucus that we have here is there to help protect the digestive organ from enzymes and to ease food passage. And there are a lot of enzymes that can be secreted from there or hormones. The next part here, the lamina propria, this is generally made up of loose areolar connective tissue, and it's basically rich. Let me take this. So the epithelial has the mucus, right, to help to protect it and to help push food across. This guy right here basically has a lot of capillaries in it. And that's important because it's needed for nourishment and absorption. In this area also, you might remember learning about molt before, and that's what you find in there, right? That is that mucosa-associated lymph node tissue. So this is the area that's going to help defend against microorganisms. And then, of course, this guy right here, the muscularis mucosae, that's going to be important in movement, right? It has smooth muscle here, and so that's going to be what helps to propulse with the propulsion of the food across through there. Now, this submucosa area here, this is going to be important because it has a lot of elastin or elastic tissue here. So this is a part that helps to regain shape. So the large amount of movement that you see is going to be this muscularis externa. And this is where the brunt of segmentation and peristalsis comes from. Now this one right here, the muscularis mucosa, that is going to be the movement of allow the local movement of the mucosa area there. And basically the serosa is the outer layer. Now, when we talk about this esophagus, we're going to talk about the inventita, and that's what is the serosa is in the esophagus. But this is the basic structure. You can see the plexuses there that'll help with activating digestion. So this is the basic structure of the alimentary canal. So just to review, the innermost of the mucosa, the outermost is the serosa. The mucosa, the epithelium, is going to be where you produce all the mucus, and that's going to help protect it and allow food to move through it, right? The lamina propria is going to be the area that you have malt in. And so essentially that's going to be where nourishment happens and absorption because of the capillaries are located there. And that's going to help defend against microorganisms. And this muscularis mucosa is going to help produce local movement of the mucosa. The submucosa right here is basically going to be the area that has elastic tissue in it, so this organ can regain shape, especially after large meals. This muscularis externa is going to be where segmentation and peristalsis happens. It can even, and you see it has inner circular layers and outer longitudinal areas, so there's two different areas there. And the circular areas are actually will thicken in some areas, and that's where you're going to get sphincters, and we're going to talk more about sphincters later. And the serosa is the outer layer, right? You can actually see it's made up of the visceral peritoneum, and it's a protective layer over the whole thing. So, when we're talking about the alimentary canal and we're talking about the digestive system, how is blood supply handled? And so you have both what's known as the splanchnic circulation and the hepatic portal function. And so basically with the when we need the digestive system to actually get blood, we're going to go through the splanchnic circulation. So this is to serve the digestive system. The hepatic portal circulation is a little bit different, right? On here, its function is the drain. So it's going to drain nutrient-rich blood from the digestive organs. And I'm going to put DO for those, right? And it's also there to help deliver blood to the liver so that they can process it. So the blood supply is a little different, right? We need a blood supply to get to the digestive system, to serve it, to give everything that you need. So that's where you have the splenic, the left gastric arteries, and the hepatic arteries giving blood to those organs, along with the inferior and superior mesenteric arteries. So those are all branching off the aorta, and it's to serve those digestive organs so that they can have adequate supply of oxygen, adequate supply of nutrients. Now, the hepatic portal circulation is going to be whenever we get done and we're basically trying to put that blood back into circulation, what is it doing? And so in that case, that's where you're going to take that nutrient-rich blood from the digestive organs, and it's going to drain it and deliver it to the liver to process it. So then once everything is removed that needs to be removed from that, it can be recirculated into the body. Now, how do we control the digestive system? And we were talking earlier that we had two basically, enteric nervous system, and we have both a localized system and a overall system that helps with it. And so the enteric nervous system is what we're going to talk about first. So basically the GI tract has its own nervous system. It's called the gut brain. It contains more neurons than the spinal cord does. It's made up of enteric nerves that communicate extensively with each other. And so the major one second, guys. So the major nerves supply to GI tract wall that controls basically motility. Okay. And so the enteric nerves make up to the bulk of two main interconnecting intrinsic nerve plexuses. We have both what's called the submucosal nerve plexus, and then we have the myenteric nerve plexus. And so the first regulates glands, and then the smooth muscle in the mucosa. And the other controls the GI tract motility. So now let's look and see where these different plexuses arise within the different layers. If you can see what we talked about earlier, right? The mucosa was here. So that's the epithelium that we talked about was important in making the mucus, right? And then this had the malt, so it protected them. This had the smooth muscle that essentially helped to move mucosa, right? And then you have the submucosa. And the submucosa was important because it helped to basically get the shape back, right? And so this is that first plexus, or that submucosal plexus that we were talking about, that helps to regulate glands and smooth muscle in the mucosa, right? And so it is located in the submucosal area. It's also known as the Meissner's plexus. The Auerbach's plexus, or the myenteric plexus, is located within the muscularis propria. And essentially, remember that the thicker circular muscles can essentially be the sphincter muscle, right, or the sphincters that help to separate the two. And this is the area that's responsible for segmentation and Peristalsis. So this should make sense that this is located here because this is the plexus that controls GI tract motility, right? So if you think about what each of these plexus does, it should be very kind of forward that these are the areas that they're in, and this is the outside, right? And so this is the protected area here. That essentially is comes or is part of the visceral peritoneum. Okay. So that kind of shows you how the general organization of the gastrointestinal tract is and where those different plexuses are that help to basically communicate with that gut brain. And these are part of the intrinsic nerve plexuses, right? So now when we're talking about the enteric nervous system, also participates in both short and long reflex arcs, right? And so there's a whole part that's kind of part of both happens immediately and is quick and in the area, and that's a little bit farther out. And so short reflexes are mediated by the basically enteric nerve plexus, which we just talked about, right? And that's the gut brain. So the short reflexes, think it's short, so it's going to respond to stimulus that's actually in the GI tract. Now basically the long reflexes can respond to the stimulus inside or outside of a gut. So this can come from the autonomic nervous system. Remember rest or relaxation, fight or flight? So parasympathetic system is going to enhance. So this is rest, relaxation. This is going to be, there's not a bear that's going to be chasing you. So at this point, this parasympathetic system that's coming from the autonomic can help to enhance digestive system to help get those molecules that you need to build things. Sympathetic nervous system is going to be something like a fight or flight, and this is going to inhibit digestive system. You don't really need to digest food while you're running from a bear. And so that's basically how our system regulates digestion. And so here you can see just a nice cartoon of how it works. So if we're looking at an internal stimulus here, this is the lumen of an alimentary canal, right? And so here in the internal GI stimulus, it's going to be something that's going to activate a chemoreceptor, an osmoreceptor, a mechanoreceptor, something that's there. And basically the short reflex is going to activate the intrinsic local nerve plexus or the gut brain. And so what happens here is it can activate a smooth muscle or gland. And when that happens, you can get either secretion from the gland to basically start with the chemical digestion, or you're going to get change in the contractile to start with the basically mechanical digestion. Now, the internal stimulus can go also to a long reflex, which means this is going to be something that has to do with the central nervous system here in this long reflex, right? So the visceral afferent is going to go away and talk to the central nervous system That can also be an external stimulus. So if you see, smell, have any kind of taste or thought of food, that can also talk to the brain, right? And then the autonomic nervous system is going to come in and basically talk and allow for a smooth muscle or gland to also be activated as well, right? And so the long reflex, just think for the most part, it's going to be the autonomic nervous system that's going to either turn it, increase it or decrease it, right? And that's going to be long reflex because it's going to go out of the actual alimentary canal away from the digestive system to decide what's going to happen. Where the short reflexes are going to be basically the gut brain, or one of those intrinsic nerve plexus is going to decide what's going to happen. So now let's talk about all the different organs. So let's start first with the mouth and the associated organs. So first, we're going to talk about the functional anatomy of the digestive system. So mouth is where food is chewed and mixed with enzymes containing saliva. That begins the process of digestion, and swallowing process is initiated. So the associated organs included to this is the mouth, the tongue, the salivary glands, and the teeth. So let's talk about the mouth first. Sure, I'm on the right. And so, ingestion occurs only at the mouth. Okay. It's bound by lips anteriorly, cheeks laterally, a palate superiorly, and tongue inferiorly. And we're going to look at all this, right? The oral orifice is the anterior opening of the digestive system. Walls of the mouth are lined with stratified squamous epithelium, and that's important because these cells are tough, so they resist abrasion. Think about how many times you've bitten on the side, and the kind of food that you bring in sometimes is very sharp or pokey, and so every time you bring food in, you're not essentially targeting yourself in the sense that you're going to bleed out through your mouth. So they are pretty resistant to not being broken through. Cells of the gum, hard palate, and part of the tongues are keratinized for extra protection. So they do have keratin, that protein keratin in them, that's going to make them way more robust. And so as you can see, this interior part, or this anterior view of the mouth, right? You have the lower lip, and you have the upper lip. And then if you look, you have this kind of separation here that's in the middle. This is what's known as an inferior labial frenulum. And then you have the one on the top that's a superior, right? And then if you look, you essentially have the oral vestibule is that area that's under your tongue, that's between your gum line, which is the gingivae, and your lip, right? And then you can see anytime that you have these lines that are connecting them, the frenulums, you can see another one of those underneath, and that's the lingual, right? And that's going to be there kind of helping to connect this bottom area to your actual tongue, right? And you can see these areas right here are the sublingual folds. that are going to have the sublingual ducts. And then down here you can see the little openings of the submandibular ducts there as well. Let's see. What do I want to do on here? You can see at the top here, you have the hard palate that is on the top there. And that hard palate is basically formed by the palatine bone and that palatine process of the maxilla with a kind of this midline ridge up there. You have the soft palatine that comes down here. Mostly those are folds of skeletal muscle, if you're wondering. And this guy is going to be what ends up closing off the nasopharynx during swallowing. This area, that's actually these guys that come up and you see those two arches. So the you can see that comes down as the uvula, right? And let's see if there's anything else that I want to do in here. Sorry. You can see the tonsils back here in this area. The palatine reef is this area that's connected between the two. Basically helps to, that's that midline ridge in the hard palatine area. And I think we're good with that for now. Now, if we're looking at the side here, you can see, and so, let's see if we're going to talk. Let me look forward really quick. Yeah, I'll talk about the salivary glands here in a minute. And so you can kind of see this back area. So when we're talking about, you can see the tongue here. And so this is the oral cavity that is kind of this mid-sagittal cut that you're getting. So you can see the oral cavity here. You can see the uvula going down here. Of course, we're going to talk about all the different salivary glands. But if you notice, you have this area that's back here, which is the orthopharynx area, and that's the area that's going to be cut off and separated so that swallowing can happen, right? And then you can see down here in this region is the laryngeal pharynx, which we're going to talk a little bit more about when we get to breathing. And so, and then of course you have the trachea here and the esophagus here. And basically we're going to have the epiglottis that will cut this off here so that everything can go down there versus going down to your lungs. And so this is kind of more of that kind of general anatomy of the mouth on here. And so when we're talking about the mouth, right, a big part of the mouth is the tongue. It occupies the floor of the mouth. And basically, it's composed of bundles of skeletal muscles. And it has basically three functions. It's important in gripping, repositioning, and mixing of food. And this happens during chewing, right? It's important in the formation of the bolus. And so the bolus is this mixture of food and saliva. And it's pivotal in the initiation of swallowing, speech, and taste. Now, the muscles that are important in changing the shape of the tongue are the intrinsic muscles. And the ones that help to alter the tongue's position are the extrinsic muscles. The lingual frenulums are attached to the floor of the mouth, and that helps to kind of stabilize them. So let's look at the papillae that are visible on the tongue. And there's different ones, right? So the first one that we're going to check out here are these ones here. We have the filiform papilla. So these guys right here are important. And what these all are these peg-like projections. And they're kind of this underlying mucosa that are in the mouth, right? So this gives the tongue roughness. Actually, let me just put roughness, right? So that roughness is going to provide friction. This is not a taste bud. And this can give the tongue a whitish appearance. The next one that you can look at here is the fungiform papilla. This guy is mushroom shaped. It's scattered all through the tongue. This is what gives the tongue the reddish appearance. The valate papilla, also known as the circumvallate papilla. These guys, they're about 8 to 12 V-shaped rows. And they're on the back. And the folate. You can see those right there. Papilla are located on the lateral aspect of the posterior tongue, right? You can see them over here on the side. Now, if you look on here, guys, you can see this guy right there, the terminal sulcus. That's right back here, right? So this guy actually marks the division between what's considered the body and the root. And so the body is the portion of the tongue that's in the oral cavity. And the root is the part that resides in the oropharynx. Now this guy does not contain any of this papillae. It seems rough, but that's because of this tonsil, the lingual tonsils back there. But it doesn't have any of those. So those are the different parts of the tongue, or at least those peg-like projections. Now, within our mouth, we're going to have salivary glands that essentially produce saliva. And the function of saliva are many. For instance, it helps to cleanse the mouth. It helps to dissolve food chemicals for taste. It moistens food. So it basically makes a bolus. And it begins the breakdown, specifically of starch. And the enzyme that does that is amylase. So most of the saliva is produced by the major extrinsic salivary glands located outside the oral cavity. Minor salivary glands are scattered throughout the oral cavity, augment slightly. So let's look at those salivary glands. So just to give you an idea, saliva is mostly, let me see. Saliva, is mostly water, okay? But it does contain electrolytes, amylase, specifically salivary amylase, right? What's known as lingual lipase. It's going to have mucins, lysozyme, and antibodies. And of course, there's always some waste in there, metabolic waste, right? So when we're talking about salivary glands producing saliva, we have a couple. We have basically a few that we're going to look at here. You can see the very first one is this parotid gland. You can see it's up here. Here's the masseter muscle. So it's back here towards the back of the ear. Submandibular gland. Think about the mandible towards the bottom. That's going to give you an idea here. And sublingual, which is around the tongue. So those are the three main types of salivary gland, and that are going to be basically producing saliva. Now, when we talk about the production of saliva, it's basically salivary glands are composed of two main types of secretory cells. There's serous and mucus. And so when we're talking about what saliva is made up of, the serous actually produces the watery secretions, the enzymes, the ions, and a bit of mucin. The mucus cells produce mucus, right? So when we're talking about those parotid and submandibular glands, contain mostly serous types of cells. So underneath, towards the back and towards the mandible, basically they're going to be producing all the watery secretions, enzymes, and all of that. And the sublingual, which is closer to the front, right? It's going to catch most of the bad stuff. Is the mucus, right? So think there, it's towards the back, or it's closer to the front, right? So it gets the bad stuff. These guys, closer to the back, digestion. That can help you remember the two. So just a reminder, saliva is mostly made up of water, so it's hypoosmotic. It's slightly acidic. It has electrolytes in it. It has different types of enzymes, salivary amylase and lingual lipase. It produces proteins like mucin, lysozyme, and IgA. IgA is another nice way of just saying antibodies in general, right? It has some waste. And just remember, there's all kinds of stuff in here that we've talked about previously to help against microorganisms, because that's an opening inside our body. So they're going to have to put some kind of defense mechanisms to make sure that we're not letting anything harmful to us in through that way. So how much do we, when we're talking about salivation in general, how much do we produce? Well, about 1,500 milliliters per day, or 1.5 liters, right? Basically, you have minor glands that will continuously work to keep the mouth moist. And the major salivary glands that we talked about are activated by parasympathetic nervous system. And those are activated when basically you have ingested food. Right? When you have that ingested food, they're going to activate either chemo or mechanoreceptor. And they, once that happens, they like to talk to what's known as a salivatory nuclei in the brainstem. And this activates the parasympathetic, which means increased digestion. And so you can get xerostomia if you have a strong sympathetic stimulation, which means if you're going to have a strong sympathetic, that's going to inhibit digestion. So that essentially will inhibit salivation and can produce dry mouth. Now the problem is if you're not making saliva, you're also not making those molecules or releasing those molecules to help to basically clean out your mouth and help to promote getting rid of bacteria. So it can produce or promote tooth decay. And what's funny is we know sight, smell of food, or epsite GAA can act as a stimuli as well. So teeth, they perform mastication, and that's basically chewing in that sense of performing mechanical digestion. Dentation and the dental formula. So basically you have primary dentation, which is talking about having baby teeth. So you have 20 deciduous teeth, which are the milk or baby teeth. And basically, they are there within 6 to 24 months of age. Then you have secondary dentation. Basically on here, you have 32 permanent teeth. And they come out and occur between the age of 6 to 12 years. All but your third molars, which are wisdom teeth. Some people don't even have wisdom teeth. For instance, my son doesn't have a set of them. They're normally in by the end of adolescence, between 17 to 25, but sometimes the wisdom teeth can come out later in your 20s, later 20s, early 30s, but most of the time it's between the age of 17 to 25 years of age. And so let's look at the types of teeth we have and point them out. And so we have incisors. These are basically those kind of chisel shaped that will help to cut food. They have one root towards the front. The canines are towards the side, and they're fang-like, and they tear, they help pierce. They have one root. Premolars, which are also called bicuspids. These are broad crowns. They're rounded cusp used to grind or crush. They normally have one root, except the first upper premolars. And then the molars are these broad crowns. They're rounded. They're best grinding, right? And your first two upper molars have 3 roots. The first 2 lower molars only have 2. And the third molars vary. You don't need to know how many roots that they generally have. I just find it interesting that they have all these different roots. So you can see, and basically they're talking about with the deciduous or the baby teeth when they come out. So basically they don't all come out at one time, and we all know this because you've been a child or you have children yourself. The central incisors are the first to come in. There's normally about 6 to 8 months. And then you'll have the lateral, which are the second at 8 to 10 months. And then basically you'll have your first molars, which are back here, which are about 10 to 15 months. Then you'll have your canines come in, also known as eye tooth, between 16 and 20 more. And your secondary molars are your last around 2 years. And then again, these all fall out and get replaced by your permanent teeth. So as you can see with these guys, it's roughly about the same. These guys come in first, right? So your central incisors come in around the year 7. And then you get your lateral that will come in. You'll get your first molars coming in around 6 and 7 as well in the back. You'll have your next coming in are your canines around 11. Your premolars here around the second time. And then all of these kind of do the second premolars, your second molars, And then these guys at the end are last, if they come in at all, or if you even have them, which are your wisdom teeth. Again, don't need to know what year they come in, right? What's important really here is what their functions are, and can you identify where they're located in your mouth. And so this kind of gives you an overall idea of what the tooth structure actually looks at. And once we're done with teeth, we're going to take a break. And so each tooth has two major regions. You have the crown region, right? And then you have the root region here. And so the crown is the exposed part that's above the gums, right? It's covered by enamel, the hardest substance in the body, right? And so this guy is very much has heavy mineralized with both calcium salts and what are known as hydroxyapatite crystals. And we talked about this when we were talking about bone, right? So enamel is not replaced. Enamel producing cells actually degenerate when tooth erupts. So no healing if the tooth decays or cracks. You need artificially replaced by filling, right? We know this. And so you can see the enamel here, and then on this inside, basically you'll notice the dentin here. The dentin is a bone-like material, right? It's maintained by, you'll see that you notice that you'll have the pulp cavity that's right here, and you have what's known as odontoid blast. Think blast cells and orthodontists. Odontoid blasts are maintained, will maintain that dentin or bone-like material underneath the enamel. Now you have the pulp cavity. This is where the blood vessels and the nerves are. And then basically, within that is pulp, right? And this is just connective tissue, blood vessels, and nerves. And then you can see as it goes down, you have the root canal there that goes down to what's known as the apical foramen, right? This is on the proximal end. And this is the entry, right, of basically blood vessels. I'm going to put BV for blood vessels and nerves. Now you can see from all this is part of what's known as the root. And so this is the portion that's actually embedded into the jawbone. And these guys are connected to each other by the neck. So on here, if you remember, the periodontal ligament is here. And if you remember, what kind of fibrous joint was this? Hopefully you remember the gamphosis joint, right? And so this periodontal ligament that's there actually anchors the tooth in that socket, right? And basically this, what connects the periodontal ligament here to the actual root is the cement. And the cement is just calcified connective tissue. And so that kind of gives you an idea of the teeth and what the teeth are important. So at this point, I'm going to take a bit of a break. since we are now done with part 1 of this lecture, and we will come back to finish this with part 2.